Good afternoon, everyone. I'm Mike Dombeck. I lead corporate development for Precision BioSciences. Just to remind everyone, please keep in mind I will be making forward-looking statements during this presentation. So Precision is a genome editing company dedicated to improving life through our efforts to create a more sustainable food supply, cure genetic diseases, and overcome cancer. At the heart of Precision is a team of over 180 precisioneers led by a group of pioneers in the genome editing space, working with our unique and proprietary genome editing platform we call Arcus to deliver allogeneic off-the-shelf CAR T products, more nutritious, more sustainable foods, and our in vivo gene correction therapies. Precision's near-term development strategy across all of these platforms is to lead with focusing on validated targets, building out capabilities, including our own in-house manufacturing that we can leverage across these different platforms, while complementing that by leveraging our industry-leading partnerships across each of these business lines to add downstream capabilities to our work. And then positioning follow-on programs to advance rapidly in each of these business lines upon proof of concept with the lead program. Our core technology, Arcus, is the only gene editing technology that is derived from a naturally occurring gene editing enzyme. And it features 4 key attributes. Number 1 is safety. Arcus self-inactivates in the absence of its intended target. What this means is that we don't get random off-target editing. Second is delivery. Arcus is 1/5 the size of the next smallest gene editing technology. This significantly facilitates delivery either via viral or non-viral delivery vehicles. Third is precise control of edits. Arcus is unique in that it leaves a 4 base pair 3' overhang when we effect an edit, which gives us the ability to effect any type of edit that would be desired. And finally, Arcus was developed entirely in-house at Precision and is wholly owned by Precision, and as a result has unmatched freedom to operate in the gene editing space. Turning to our off-the-shelf CAR T portfolio, Precision is really focused on balancing and optimizing 4 key parameters for CAR T: yield, high quality, low cost of goods, and target engagement. The result of our efforts with this platform are a highly consistent product available on demand off the shelf with minimal complexity in administration and expanding the flexibility in dosing for the oncologist. The result, we believe, will be significantly increasing patient access to CAR T technology. At the heart or core of our efforts in developing our CAR T platform is an emphasis on optimizing the CAR T cell phenotype for in vivo expansion. In vivo expansion is what drives efficacy or therapeutic response with CAR T, and what drives in vivo expansion is the percentage or proportion of naive and central memory T cells in the treatment for the patient. Precision's manufacturing process consistently produces a product with greater than 50% naive and central memory T cells. We've taken this process and fully scaled it up to cGMP production of clinical trial materials. The data you see here are from actual clinical trial material runs. From a single draw, a single donation from a single healthy donor, we consistently produce a minimum of 100 doses of our CAR T products that are greater than 99% CD3 negative, 65 to 75% CAR positive, and again, importantly, always greater than 50% naive and central memory T cells. Combining all these things, we believe we have a best-in-class product profile for an off-the-shelf CAR T program, combining a rapid single-step manufacturing process, a proprietary co-stimulatory domain, our optimized cell phenotype, and a more mild lymphodepletion, we believe that we'll have a superior off-the-shelf CAR T product. And we're deploying that profile across multiple targets. Our lead target is an anti-CD19. This is currently in phase 1 clinical trials. Will be followed by our anti-CD20, which we recently received IND clearance from the FDA to pursue. And our BCMA program, we anticipate filing an IND in early 2020. Turning to our CD19 program, as I mentioned, this is in a phase 1 trial currently. Our first patient was dosed in April of this year. We are pursuing both NHL and ALL, and we were the first to dose an adult NHL patient with an allogeneic product. We will be presenting interim data from this no later than first quarter of next year. As I mentioned, this will be followed by our CD20, which recently received IND clearance from the FDA. It also received orphan drug designation. We anticipate initiating this trial before the end of the year with interim data from this program presented in 2020. And finally, as I mentioned, the third program behind this will be BCMA with an IND early in 2020. Turning to our in vivo gene correction platform, here we've emphasized 3 key parameters: ensuring the safety of our in vivo gene correction approach, delivering efficiently both viral or non-viral options depending on which therapeutic targets we're pursuing, and ensuring precise control of the edit that we're affecting. The result are in vivo gene therapy candidates that eliminate random off-target cutting, that enable delivery to most tissues, where we are able to affect any type of gene edit efficiently, and with complete control over the use of the Arcus platform, the ability to maximize the scope that we can approach for in vivo gene correction. Our portfolio here is led by our Hepatitis B program, which is partnered with Gilead. Behind that, we have a number of targets across the liver and the eye. All of these targets are currently in large animal models, and based on the results of the large animal work that we do, we anticipate selecting a lead internal candidate before the end of this year. Turning to a handful of these programs, first, the Hepatitis B program is one where we deliver an Arcus nuclease to disrupt both integrated and episomal hepatitis B viral DNA in the liver. Again, this is a partnership that we're working on with Gilead, and we anticipate the IND for this program towards the end of 2020. On some of the internal programs we're working on, this is data from a PCSK9 knockout that we did with Dr. Jim Wilson at the University of Pennsylvania. This program was published last year. It was the first publication of a therapeutic gene editing knockout in a non-human primate. As you see here, the first cohort of animals in this is now out past 2.5 years with a permanent therapeutic edit, essentially eliminating PCSK9 and reducing bad cholesterol by 50%. So sustained permanent efficacy with no toxicity or tolerability issues. The last program I want to touch on is our autosomal dominant retinitis pigmentosa program. This is a program where we use an Arcus nuclease that selectively eliminates or disrupts the mutant allele, the dominant mutant allele, while leaving intact the silenced wild type allele. This program, again, is in large animal models right now. The relevant model here is a pig model, and we have demonstrated that we are able to restore photosensitivity in these animals with a single administration. Over the course of the next year and change, we anticipate a number of near-term value catalysts. You see a number that we've already delivered through 2019. Coming up next is initiation of the CD20 CAR T trial before the end of this year. Interim data from the CD19 trial no later than the first quarter of next year. filing our IND for our BCMA candidate early in 2020, and then finally our first in vivo gene correction IND before the end of 2020. And as you see, our cash runway takes us through all of those milestones and into 2021. So in conclusion, some key takeaways. Precision brings together a highly experienced team, over 180 precisioneers with some of the pioneers in gene editing, working with the proprietary Arcus platform with which we have full freedom to operate. We've brought online our own manufacturing capabilities, and by the end of this year, we'll be producing our own clinical trial material in-house. Multiple allogeneic programs that will be in human clinical trials before the end of this year. Our initial CD19 data, which will be presented no later than first quarter of 2020. And finally, a strong balance sheet combined with our validating industry-leading partnerships that support each of those business areas. Thank you for your time. I'd be happy to take questions in the back of the room afterwards.